My name is Jing PAN, come from Beijing GoBroad Hospital in China. Now I want to introduce my story and GoBroad story and how to do the closed loop of clinical translational research in T-cell malignancies. First, I want to introduce our hospital. Beijing GoBroad Hospital is a research hospital in Beijing, and we focus on the cancer immunotherapy, clinical research, and we also have the B+A grade GMP lab. And now we provide 40 cases of point-of-care CAR-T manufacturing per month in our hospital. And here is our, and the picture showed our ward, and we had 18 beds in our group, so for the clinical research. And what about us? I'm the PI in our group, and we had a focus on the CAR-T cell therapy in hematological malignancy for 9 years. So in the early 5 years, we focus on the BAL. So we do the CD19 CAR-T products, and we found some problems. So we proposed the sequential CD19 and CD22 CAR-T cell therapy structure for them. And since 2019, we focus on the T-cell malignancy. So we do the CD19 CAR-T, CD7 CAR-T cell clinical trial and the CD5 CAR-T clinical trial. And in the early 5 years, when we do the CD19 CAR-T products, we found some problems. The major problem is the relapse. So we focus on resolve the clinical problems. So we want to do the change to reduce the relapse. So we proposed the subsequential CD19 and the CD22 CAR-T strategies. We do the first one and the first two clinical trial, and then we found it works. So we also have many Chinese family, and they had more than 9 years cancer-free in our group without transplantation. So since 2019, we changed the area. We focus on the T-cell malignancy. What we do in the past 5 years, we established two steps. And the first step is 3 platforms for avoiding CAR-T virtual sites. Here, we had 3 ways to do the. We can do the CD7 lockout. We also can use the retention domain to track the CD7 in the ER. And we also can do the natural selected to avoid the CAR-T cell virtual sites. And then we also want to choose the ideal T-cell sources. So we had 4 platforms. One is for autologous CAR-T cell product manufacturing. So the data had been published last year. And when the patient didn't have enough T-cell for the CAR-T cell manufacturing, we do the donor-derived CAR-T cell. This clinical data has been published in JCO. And recently, we even use the cord blood. When the patient didn't have the donor, they also didn't have the enough healthy T-cell, we use the cord blood. We use the cord blood to the manufacturing, and it works. So we established 2 platforms, and we focus on the T-cells manufacturing, and after that, we do the clinical trial. So our group are the leading sites in multiple center clinical trials for this area. So this table had shown the ongoing clinical trial in this T-cell malignancy area. We can find the first one here. We can find the less than 1 year, the increment had been finished. So the increment rate is very high in our group. And now we focus on the CD5. Why is the CD5? we think there are large numbers of patients need CD5 CAR-T products than CD7. So we only can use the CD7 CAR-T products for the T-ALL patients, but we can use the CD5 CAR-T products for PTCL, DLBCL, CLL, and TAL patients. So we trying to cooperation with the biotech. So this is the example for the cooperation with the biotech. The ISO biotech had a authorization their vector sequence to us. After that, they do nothing. Everything had been done by our group. The car vector production, GMP manufacturing, and clinical trial design. And we also conduct the clinical trial. So the data had been published on Nature Medicine last year, and we had amazing and high quality of clinical data of this proof of concept. But it showed new program during the translational research. We found we need an ideal expansion time for the CD5 CAR-T products and the CD7 CAR-T products, because we need the CAR-T cell expand quickly more than the tumor progression. And the other program is the on-target and off-tumor effects. We can find this CAR-T product can induce the autoimmune disease. So what's the next step for us, our group? First, we trying to find a new CAR vector. So we screen the new nanobody-based CD5 CAR vector. Here we can find the green line. Green regions show the data had been published in Nature Medicine. This CAR-T can bind domain 1 and domain 2, but we find a new vector. This vector here can bind domain 1 and domain 3. And after we had finished the preclinical data, we fast testing in clinic. So only need one month, we do this fast testing, proof of concept. We involved the patients that had a combined relapsed bone marrow and extramedullary disease and a high expression of CD5. And we also do everything in our group in GoBroad Hospital. And after that, we found an ideal AE, and the peak time of the CAR-T cells is day 11. It works. So after that, we conduct a multiple center clinical trial. You can see the NCT number in the NCT wipe, and this clinical trial is focused on the PTCL in China. We will publish the data. We should publish the data in next ASCO. And also had a response for the extramedullary disease in this new CAR vector. But more solution on the road. We want to control this CAR-T. We want to eliminate the on-target and off-tumor effect. So we need a smart CD5 CAR-T. So that's the GoBroad model and the closed loop of translational research. So I also welcome to all of you to enjoy us at any part of the loop. You can enjoy us on the basic research or the proof of concept, even for the IND permit. So I will stop here, and I want to very happy to take questions.